think we can start now. Good morning and good afternoon to everyone who's joined. I'm really excited to see that so many have to participate in this webinar. First, some practical information. Everyone is muted throughout the webinar, but you are more than welcome to ask questions through the Q&A box. You can find it here on the bottom of your page. I'll be your host for this webinar. My name is Julie Vistisen, and I'm the product manager for CelVivo. I have a background in molecular biology. Today's webinar is titled Why Active Diffusion and Low Shear Forces Make a Difference when Cultivating Cells in 3D. I'll go through some of the basis for cell culture models and what you should consider when cultivating cells in 3D. And then also the challenges of going to the third dimension. And I'll introduce you to a new technology and share some of the applications that you can use. First off, when you're doing research, you need a reliable model for your research. And common models that has been used are 2D cell culture models and animals. And they're really good models because they fulfill a lot of the requirements that you have for a model. However, you want a good model to resemble the reality. So if you want to study a human, the ideal situation would be that you had human samples. However, that is not always the case. So you want your model to be as close as possible. You also want the model to be reproducible. The less reproducibility you have between your models, the more you can trust the data you get out. And of course, you want your model to be very abundant. Like when you're going to the store, you want all the groceries to be there, and you want to be able to get as many as you can. So why should you consider going away from these mice or cell culture models? Because they are both reproducible and very abundant. The problem is that you want to study humans or human cells. They don't really resemble the reality. And even if you wanted to use 2D cells to study animals, that's not the case that they resemble the animals that they are studying. So we have some challenges. And that is why 3D cell culture has emerged as a popular technology. And there is numerous published material that says that 3D cell culture predicts the situation much more accurately than normal 2D cell culture models or even mice. So let's have a look at some of the 3D cell culture characteristics. And today I will focus on human samples. So we have this human here in the center. And of course, a human sample is heterogeneous. So if you take a look at a liver, for instance, it's going to be different. The cells are going to experience different nutrient and gas exchange on the outside of the liver than on the inside, because there is going to be diffusion gradients between. So a good sample should be heterogeneous. And that's also what we have in 3D cell culture, where you cultivate these cell aggregates and the cells in the middle are experiencing different nutrients access compared to the cells on the outside. So in reality, you have not different kinds, but your cells are heterogeneous. As in humans, we also see that cell cultures or 3D cell cultures have a decreased proliferation rate. So they don't proliferate very crazy like cancer cells, for instance, would do. But their proliferation rate slows, and it resembles what's going on much more in vivo, where your tissue do not proliferate all the time. It should also have relevant genetic markers. So if you wanted to study the liver, it should have relevant genetic markers to the liver. And it should also have relevant secretion levels. And furthermore, it should look like the tissue you want to study. So the ultrastructure should resemble the tissue you want to look at. And then it should have some complex cell interactions. So the cells are not just they speak to each other, and these cell interactions, they need time to reestablish. And furthermore, it should also predict the situation in vivo. So when you're cultivating cells in these 3 dimensions, there are 2 things you should consider. One is that you need this structure to form. You can't just form a monolayer of cells. You need the cells to grow in a shape in the 3rd dimension. And then you need to secure that there is enough nutrient supply to these cells, because there's thousands of cells conglomerated. And you have to consider this when cultivating. But we're going to start to look with the structure formation. So when you want to achieve this complex ultrastructure of cells with cell communication, you always start the same thing. You start with a sample, and it can be from tissue or from a cell culture. You decide. And you want this sample of single cells to conglomerate into a more defined and complex structure. And there are several ways to do this, and I'm going to mention some of them. Some cells are reliant on this extracellular matrix to form spheroids or organoids. So cells can be cultivated in this gel-like matrix or scaffolds. And when the cells are cultivated in this, they get the time they need to reassemble and conglomerate into 3D cell structures. You could also use plates. That's a very popular technology as well, where they have an ultra-low adherent surface. So when you put your single cells in the plate, they will also conglomerate into 3D structures. As I said, these are only a couple of technologies, but they are very popular and emerging because they're really good at forming 3D structures. However, there is a problem with these, because how do you make sure that there's enough nutrient supply for your cells? In these things, they're very static, so you don't have any movement of the media. And the direct consequence of this is that your constructs will experience passive diffusion. So it's a challenge, and it's a diffusion bias, because you want your cell constructs to have access to all the nutrients that they need. And because you just added a 3rd dimension and a 3rd complex or more complexity to your cell constructs, there's going to be longer to the center of your cells. And if you don't have if they don't get the nutrition they need, they're going to suffocate and die. So in this situation, you want active diffusion. And active diffusion is secured when the media is moved around the construct. And you can imagine that when the cell is standing still, it will start to eat whatever is around it, and it will use the gases around it as well. But if you continue to exchange this, the cell will get the nutrients and get the gas and get the metabolites removed that it has to. So we have kind of created a diffusion bias for ourselves in a situation where with the plates and the hydrogel, where you get the necessary time and the cells get the peace and quiet to grow, but they don't get the nutrients they need. So if you choose a passive diffusion culture, you're going to have a problem at some point with longevity and size of your culture, because they don't get the required nutrient and gas. However, if you have active diffusion, then you can get longevity and larger size constructs. So it seems simple, and we should just go from passive diffusion to active diffusion, obviously. And people already realize this, and a lot of scientists are using various methods. One could be these spinner flasks. They are quite popular as well, or you could use a rocking table. It depends. The problem with this is that these are very rigid, and they introduce some shear stress into your cultures. So these fine cultures that you now have, the single cells that you wanted to get to reestablish and build these complex structures, now you're actually tearing them apart. You're not giving them time, and you're not giving them the peace and quiet that they need to reassemble into complex tissue. So when you use these spinner flask or other types of conglomerate or spinning devices, you're going to compromise your cell conglomeration, and you're going to compromise your reproducibility because it's an uncontrolled movement and tearing of cells. So how do we get to a place where we don't have any shear forces, but we still have active diffusion? NASA actually made use of a technology for this, because they wanted to study cells and how they would grow in a microgravity environment. So they made use of this clinostat principle. In a clinostat, it is a motor that is spinning around at a controlled speed. Usually it's around 2 to 30 RPM when you're using it for cell culture. And the benefit of this is that the cells that you see here in the middle, denoted 1, they're in suspension. And then by slowly balancing the rotation, these cells are actually going to experience that they are standing still, or they're in a statical orbit. So the shear forces you introduce into these cultures is very low, but you still get the benefit of the active diffusion because the media is slowly moving around the constructs. So it's much like the hydrogel, where the cells are basically standing still and getting time to conglomerate, but they are experiencing active diffusion, but without having any scaffolds around them. And by using this technology, we have created this new clinostar system, which is consisting of this CO2 incubator it's a standalone unit, and then it's controlled via the tablet. Inside the clinoreactor, you can make room for 6 of these bioreactors, as you can see here. The clinoreactor, the bioreactor, where the cells are going to grow, they grow in this center chamber. That's a 10 milliliter cell culture chamber. And as you can see, these blue beads, if you notice these, they are water beads. And because in cell culture, a major source for contamination in the incubators are often the water within. So we have contained the water in these clinoreactors to make sure that infection is limited. The clinoreactor functions much like a petri dish. So you can open it, and then you can access your culture. If you notice here, the material is really clear. It's polypropylene and polystyrene, so it's see-through. And over here on the opposite side in the clinostar, you can see these small black dots. They're actually cameras, so you can monitor how your culture is growing without having to open the system. And here, our founder Christoph is showing you the benefit of this petri dish opening. So he has cultivated some, in this case, spheroids, and now he's taking a few for sampling, and he can easily take them out, close the lid very, very fast. This video is a bit fast, but it still goes. And how you work with this system is actually much like a normal 2D cell culture workflow, where you cultivate your cells or your 2D culture, or you collect your biopsies or the sample you want to use, and then you select an aggregation method. And it can be, for instance, single cell suspension that is transferred directly to the clinoreactor, or you can use these micropattern low adhesion plates, or you can use hydrogel droplets. Which one to use depends on the nature of your sample and what you want to achieve. But once you have chosen your aggregation method, you transfer your aggregates or your single cell suspension to the clinoreactor. The clinoreactor is incubated in the clinostar. And with our experience for cell lines, it usually takes 18 days for them to mature and regain their functionality. It can be a shorter time period with primary cells that already have this functionality and just need times to reestablish after trypsination or removal. And during these 18 days, You exchange media every 2 to 3 days like you normally would. And obviously, as the cells grow, you need to adjust the speed to counterbalance this gravity so they are still in a static orbit. And then you also split your culture. And if any cells are conglomerated together, you remove the conglomerates in a process we refer to as gardening. And if you remember this slide I was showing you earlier, it is actually our founders who has used the system to create a liver model. So all of this data I've shown you here is from our liver model. And now I'll focus on this accurate prediction. So Christoph and Steve, they're toxicologists, and they have used the system to create this liver model, and that was their intention because they wanted a better model for their own research. And they have published numerous studies characterizing this liver model. One of them is this study where they have looked at the 3D CelVivo liver model and compared it to primary hepatocytes and a novel 2D cell culture. The 2D cells here and the 3D cells are actually the same type of cells. But you can see when you compare the in vivo lethal concentration of these 6 common APIs, and you correlate it with the LD50 value of these drugs, in a normal 2D cell culture, it does a really poor job in predicting the in vivo plasma concentration. So it cannot be used to predict if a drug is toxic for a person. Pharmaceutical companies already realize this. So obviously they're not using it, they're using primary cells. And you can see primary cells, they are the ones in the middle here. They do a very good job in predicting if a drug is lethal for a person. The interesting part becomes when we have cultivated these 2 cells in the clinostar and made them into 3D constructs. The prediction is as good and actually a bit better than the primary hepatocytes. So Steve and Christoph has definitely created a model that predicts the reality of what they studied here. And this is not the only thing that the system has been used for, because when people have seen this and they saw how it was to use, Christoph and Steve was asked if they could also get a system. So they started building systems for research collaborators and other labs in the world. And that's when things got interesting, because we've seen that this system is not only applicable to toxicology, but it can also be used for basic research and to create bone model that has been used for protein discovery. And it can be used to study epigenetics much closer to in vivo than you normally could with a 2D cell culture. It had also made some disease models. There is a diabetes model and a cancer model that predicts the situation much more accurate than a normal model would, but you still use normal cells. So you don't even rely on animals. And then of course, they have used it tremendously for toxicology. And the good thing about this system for toxicology is that you could, of course, use it for drug discovery, as it predicts the situation very well. But you can also repeatedly treat these spheroids or organoids with a drug and then look at the outcome. A situation that isn't possible before because the cells would die. So you can study accumulated toxicity as well. But I think what we can see here is that Christoph and Steve and our imagination has been, we're blown away about the possibilities of this system, and it's not only contained to one application. It's really for numerous studies. So if you think that this could be exciting for your lab, we have just launched this clinostar grant program. And what you need to do is you can provide us with an overview with your aim of your project, and then before the deadline. And then you can borrow the system, the ClinoStar system, with the 48 bioreactors for 3 months to grow your own cells and do your own research. And this short presentation was all that I had for you. And we will continue with the QA session. So if you have any questions, please type them in the QA box, and I will answer them. We will just give people a few minutes to type, and then I can go through some of the questions that we already had. So the first question is if we have culturated the primary cells in the system. And the answer to this is yes. We have culturated primary bone cells, or one of our collaborators have, in the system, and it worked well. But it's still a